I would like to invite Dr. José Mauro Granjeiro. He has a master's in biological bio, biological and cellular biology. He's the coordinator in the José Coimbra Institute, and he is assistant professor in the University of Fluminense University, and he also works with regulatory institutes, and he works with bioengineering. He works with molecular biology and bone repair. Good morning to all. Thank you for the opportunity to be part of this event. That after these excellent presentations that show us very clearly of the enormous challenge that we have ahead of us to be able to understand the interactions between the nanoparticles and the consequences that they bring not only to our organisms but to the environment. I would like to comment a bit on the Nanomaterials Institute of Brazil of quality and regulation, industrial regulation, this field that we're working in, some concepts that we are working on nanoparticles, and I'm going to comment on two projects, one in the initial phase, and the other one in planning in the area that is together with researchers of the country. The Nanomaterials has now an area called the Directory of Integrating Programs and Applied Metrology of Life Sciences that has to do with standardization and the metrics, seeing the quality of products and reference of what available materials exist. The Nanomaterials is in Xerém, that is near Rio de Janeiro. It works in areas like mechanics, acoustics, electrical, chemical, optical, and in this region, it's an ample area. They will plant a directory of programs. They will make different labs will be built, and they will be health labs and structures that will be the Brazilian Center of Biological Material that will have a project with the industry, and they will study cells and also with studies that are related to patents. And this DIPRO studies, we have the directory, and we also have the structural biology division, bioengineering, biotechnology, forensic metrology, which is the best implanted because it's the oldest, and also of drugs. The basic philosophy is to incentivize the research in the frontier areas, especially getting excellent researchers to develop projects and research that have to do with methodology. And it is research that is applied in strategic areas like nanotechnology, and to act in metrology, developing materials of reference and establishing criteria for accreditation and assessment in the conformity of the area of the life sciences. We have a convenium that is signed with Fiocruz, Anvisa, and Inmétrico to assess the orthopedic prostheses and also dental implants to begin next year. We also want a big integration with eMetro because there is an interdisciplinarity with the activity that are involved, and also with the high teaching in the country, superior teaching. When we saw the other speeches and seeing nanotechnology and how to deal with these particles that are smaller than 100 nanometers, this involves the necessity of an interdisciplinarity of professionals to face the challenges of this kind of research. We need adequate instrumentation and biological models that can reproduce and correlate the effects, especially on human beings. Here, a electronic microscope image of the particle, the fiber, and the nanometric film. We have a challenge to face that the advance in the generation of products is being much bigger than our capacity of studying their effects, since cosmetic composts and clothing and makes us close to this kind of material. We have areas that have to do with packing that have nanoparticles. So we have to study the liberation of these particles depending on how they are kept. In the medicine area, our group in Rio has an important group focus in the regenerative medicine, especially in the bone repair. Many times the dimensions are such that the capacity of repair of the organism are insufficient for the regeneration of the tissue. And some materials, for example, here we have a porous ceramic is used as an element of conduction, as a repair tissue to try to help the repairing that lesion. It is a material that sometimes remain more than 20 years in the place. So there is you can relate the mechanical properties of the bone, the functionals, and the deposition of this material. For example, in odontology, dentistry, with the oral rehabilitation with dental prostheses, sometimes you have a big interface area with the bone tissue. So we should be able to regenerate this bone area with bone, so that the implementation would be on and over the bone, considering the possibility of producing ex vivo an autogenous bone. We could use the cells of the patient himself to intensify the regeneration of this kind of lesion, and avoiding the necessity of using the fragments of bones of the patient, and using absorbing materials. These substitutions would be done in a complete way. For this, we need materials that have been developed to increase the capacity of biodegradation without losing the capacity of repairing the bone. So there is a hydroxy hepatides that are modified and structured as spheres are placed in the bone defect, and when it is made in a conventional way, which is dominates today in the market, it is placed in place without its degradation for a long time. But on the other hand, the production of these spheres that were in nanoscale, they permit the total decomposition of the material and the filling of those spaces with bone tissue. We do have a favorable effect, but we have a doubt of the security for a long term, because we do not know the fate of the particles, of those nanometric particles, and the consequence that they will have with interacting with cells. With the hepatite, we have a big capacity of absorption of proteins and biological molecules that can promote unbalance between proteins and factors in the moiety. As we said before, the nanoscale materials represent enormous change when compared to conventional material scale. So the chemistry and the energy of the surface, you have penetration that is increased, relevant quantic effects, and also the common forms of this material are unknown. And for example, gold that is seen as inactive as a nanoparticle, he is highly reactive. It is highly reactive. Within the materials, different materials that are produced, some are used with more frequency, like metallic nanoparticles, carbon groups that have been being very much tested. An important aspect is that depending on the dose, any product can be toxic. So we have to define the short-term routes and the long-term routes so that we can establish if that material in a particular concentration and exposure time, if it offers risk to the individual. And we have two groups of public that are very sensitive to this exposure, that are the people who deal with these materials, be it in the laboratory or in industry, and the final user who will be in contact with this material. It's important to establish specific regulations on nanotechnology. There are a series of reports and toxicity studies based on criteria and methodology that we're not sure that are adequate for the assessment of this kind of material. There have been international organisms that have had some actions on the regulations of nanotechnology. The International Standards Organization, the National Nanotechnology Initiative, and the British Standards Institute and the Environmental Protection Agency have proposed documents for this, but they still don't have a formal regulation in this field. Many issues and questions are open concerning the research in this area, the toxicology of manufactured nanoparticulates. They have to have an exposure assessment of these materials. They have to determine the ability to extrapolate the toxicity on the manufactured nanoparticles using in vivo and in vitro tests, and also together they are going to affect the biological fate and environmental, the transport, persistence, and the transformation of these products that have a base in nanoparticles when in contact with the organism. One of the big challenges that we have when we have the study of interaction of nanoparticles with biological models is the process of aggregation. The particles can be made in nanoscale, but during its transport, it can suffer aggregation. If this process is not monitored, we run the risk of producing these materials, but when we use them finally, we can be working with altered dimensions. So we have to see the production and maintenance to have the original physical, chemical characteristics. It's important to determine the duration and route of exposure. What we saw was the model of skin model that is an exceptional tool for this, and we have to see the dosage and formulation of the tests. It's important considering the product generation that we have the capacity of assessing the materials in a form in which they will be used by the population. The finished product has to be tested to see the capacity of liberating these particles and promote this effect that can be potentially toxic. We have to also determine the biological species to be subject of tests and materials of references and standard comparisons for our internal control of the experiments. The exposition to which the body is exposed, it can be accidental or deliberately introduced. When we introduce medical devices through nanomaterials, this form is important because it can affect the results of the study. So issues like what tests are more realistic tests must be made regarding exposition, and are exposures realistics or an exposure in laboratory with high dosage. How am I going to transfer this to the daily life? And do animal subjects respond similar to humans? So these issues imply in the standardization of models in this study area of study. An important factor that we have to see is the interference of the nanomaterial in the assay models, in the kits that are going to be assessed. We have different composites that interfere with tests of LAL. Others interfere with turbidity and will promote the jellification, and this will interfere with the assessment of the particle. Current trend is the tests in vitro that are increasing the capacity of investigating a bigger number of nanomaterials with less cost and time and with no ethical issues. So we have the response to nanoparticles. They depend on the formulation of the nanoparticles, the conditions of tests at which they are submitted, the models that are of the study that are used, and the methods that are used for its assessment. In this way, our group has worked with the development of the equivalent tissues that are 3D reconstructions that contain cells that come from the skin, from the lung, from the mucus, from the bone tissue that will be exposed to the nanoparticles to trying to portray how this will work. Although it brings the difficulty to mimetize the original tissue. Another important area that cannot be disqualified is the modeling in silica. Through the tools of molecular modeling, we can establish parameters of interaction between nanoparticles and other biomolecules, and from there increase our base of knowledge about the energies and constant association constants that can occur between these molecules. In Brazil, we are involved in with the European Union project called Nanovalid, in which we search development of reference methods for hazard identification. This project is coordinated by Dr. Rudolf Reuter, and it congregates 33 institutes in Brazil, in Metro, and the group of Professor Mauro participate. The focus is the production and characterization of nanomaterials, dots, metallic, magnetics. Also a focus on the ecotoxicology and also the human health, and clearly the improvement of the detection limits for the biological effects of the nanoparticles. There are many tasks that were foreseen in this study, and we need to validate the physiochemical and biological methods and the interaction of those with the tissues. And in this moment, we are building a proposal of a network of nanotoxicology study in the country, where we're using a selection of materials that can represent adequate tools of investigation, and in vitro assays that can see the investigation of cells, that were induced to the exposure of this material, especially with Kinoma is a tool that can identify the pathways that are activated or blocked, and this is based on in the phosphorylation of thyroxine with proteins that participate in the cascade. And another important focus is the development of tissue for skin, mucosa, and lung. And also we want it to in vivo use toxicity, kinetics, inflammation, and effective reproductive effects of nanoparticles, and observe the effect in the environment. And in this process, we have institutes that are being involved in the country, and the IME, the USP, Santa Catarina, Rio Grande do Sul, Bahia, Fiocruz in Rio de Janeiro. In this process, we have seen the complexity of that the nanotechnology, that its complexity. So we need for reference materials and standardized protocols for nanotoxicity assays, so that we can speed the secure development with acceptable costs. We cannot develop tests, very sophisticated tests, but that are difficult to be implemented and are very costly. So they have to exist in such a way that the industry can generate knowledge, and through this process can assess and validating its biological safety. It's important to determine the security of products based on nanoparticles or nanostructured materials, and using reliable and replicable methodologies. This is important in our country, so that we can permit the development that is allied with the safety of the final user and of the person who will deal directly with the production of these materials. I thank you once again and the opportunity. It is a great pleasure to be here. Thank you very much.